While ruminant animals play an important role in sustainable agricultural systems, they are also an important source of greenhouse gas emissions, especially methane. Methane is produced during enteric fermentation, a digestive process whereby microbes in the forestomach of ruminants digest plant materials into nutrients that the animal then uses for the production of high-quality produce such as milk and meat. In the coming decades, livestock farmers face numerous challenges, and the development of technologies and practices that support sustainable food production while moderating greenhouse gas emissions are urgently required. More than 100 countries have committed to reducing agricultural greenhouse gas emissions and have signed up to the 2015 Paris Agreement on Climate Change. However, known agricultural practices could deliver just 20 to 40% of the needed reduction, even if delivered fully at scale. Therefore, new mitigation options are needed to ensure sustainable food production into the future. One possible approach to methane reduction is the use of lactic acid bacteria. These are gram-positive bacteria that produce lactic acid as a major end product of carbohydrate fermentation. Lactic acid bacteria are natural inhabitants of the intestine of mammals and are among the most important groups of organisms used in food fermentation. Lactic acid bacteria are readily isolated from ruminants and are currently used on farm as direct-fed microbials and silage inoculants to increase production and improve the health of animals. These organisms are well-established industrial microorganisms, economically produced at large scale for incorporation into animal feed, making them ideally suited as a microbial technology. While it has been proposed that LAB could be used to reduce methane emissions in livestock animals, research so far is limited and data supporting the concept are lacking. The METHLAB project is investigating the possible use of lactic acid bacteria as a methane mitigation strategy on farm. The aims of the METHLAB project are to test and implement lactic acid bacteria as a methane mitigation strategy. The goal is to refine current on-farm technologies such as direct-fed microbials and silage inoculants currently used on-farm to increase production and enhance animal health with a methane-reducing benefit. An intensive screening program was carried out where lactic acid bacteria from various environmental sources as well as pre-existing cultures within our culture collections were screened for bacteriocin production. Bacteriocins are inhibitory compounds that are synthesized ribosomally. To test bacteriocin potential of lactic acid bacteria, well-diffusion assays were carried out. These assays demonstrate the potency and efficacy of the bacteriocins produced. Bacteriocins function against indicator strains through permeating the cell wall and causing cell lysis. Following on from the screen, cell-free supernatants of the lactic acid bacteria with the superior bacteriocin production were incorporated into assays which tested for methane reduction in methanogens. The cell-free supernatants were tested against a range of methanogenic archaea. Rumen methanogenic archaea are the organisms responsible for producing methane, where they use hydrogen and methyl-containing compounds generated as fermentation end products during anaerobic respiration. This methane is then belched and exhaled from the lungs via respiration from the animal and released to the atmosphere. Targeting these organisms should significantly reduce the methane produced by the ruminant. Alternatively, the use of bacteriocin-producing LAB could be used to target other organisms present in the rumen that produce precursors in the methanogenesis pathway. The methanogen screening was carried out in vitro in Teagasc in Ireland and at Ag Research in New Zealand. The superior LAB strains from these tests were then selected to be tested as silage inoculants in mini silos. The strains which showed the best results in terms of reduced loss in dry matter and digestibility were then chosen for future animal trials. Large-scale silage production was undertaken in the Netherlands, New Zealand and Ireland using the chosen LAB strains. The inoculated silage was fed to cattle and sheep to confirm efficacy of methane reduction in vivo. To date, some of the feeding trials have already been conducted and data analysis is ongoing. Using superior microbial inoculants identified in this project, we aim to improve the quality of ruminant feeds, which will lead to a reduction in methane and enhance livestock production. To achieve this, we aim to monitor animal productivity, including animal weight, milk quantity and quality. Additionally, methane measurements, which are of utmost importance, are being carried out. A green feed will be used, which is a system designed for measuring methane gas from individual animals. It is our aim that these METHLAB strains will improve animal productivity while simultaneously reducing methane emissions. METHLAB will thus lead to environmental and societal benefits, with the potential to deliver green jobs and enhance the competitiveness of the agri-food sector. Projects like this are key factors in contributing to global greenhouse gas reduction. METHLAB brings together a global network of multidisciplinary researchers to enhance impact and advance technology transfer of lactic acid bacteria on farm technologies. METHLAB addressed the reduction of methane emissions in ruminants, in particular cattle and sheep production systems. The consortium is led by Teagasc and involves partners from University College Cork, Ireland, INRA in France, Wageningen University in the Netherlands, Ag Research in New Zealand and the culture company SACCO in Italy. The EU funding amounted to over €1 million and the project duration was 36 months. The partners in this proposal are from Global Research Alliance member countries that share the goal of reducing methane emission intensity across ruminant classes in a manner that maintains agricultural production and sustains environmental integrity.